Welcome to the lab again. Today I want to show us how we, at the end of an experiment, say you are having an in vivo experiment that has to do with treatment of animal models. For example, we use the mice as animal models, then we can use some primates as animal models. So for instance, if you are using say rodents like mice for your animal models, at the end of the treatment period, at the end of the treatment duration where you have administered all the treatments required, you will need to get some vessels ready. So like the EDTA containers, then you will get the um, capillary containers, the capillary tubes. So all of these will be used to get the blood and some other tissues that will be required. So you have to assemble them, have them washed and kept clean so that then they will be adequately labeled, because the animals you are using, they have been grouped initially to different groups, say group 1 to 6 or group 1 to 7, group 1 to 5, depends on it, then and the control. So meaning that all the vessels also, the capillary bottles, the capillary tubes and all that you are using, they must also have that labels on them to show the exact group. So you'll be able to group them according to their several groups. So you know that, okay, these ones are for the controls, those ones are for the group 1, group 2, group 3 and all that. So once those has been taken care of, then you can now make sure that they are covered adequately. So it now depends on it. So okay, let me quickly mention that when you are culminating your experiment, at the end of the experiment, you want to, for example, get the required blood samples or get the required tissues from the animals. You have it, you do euthanasia. So euthanasia is messy killing. So you ensure that you follow the procedure, the ethics of animal sacrificing, such that the harm is not done, overall harm is not done to the animals, and is done in according to international standards. So these are the requirements you have to put in place. So if you require decapitation, it means that you will use the surgical equipment to have the animals dissected and their cervical decapitation will be done, and the vitals will now be removed on the long run and taken to the lab for analysis. So that is why you require everything to prepare beforehand, and so that on that day of the decapitation, you will not be having yourself unsettled. So these are the things you do when your experiment is an in vivo experiment is coming to an end. So thank you. See you in the next video.